Good evening and welcome to Global Studies 287, a first of its kind community course offered by PLU. I'm Tamara Williams, the Executive Director of the Wong Center for Global Education, also a Professor of Hispanic Studies and former Chair of PLU's Global Studies program.
And I'm Teresa Chabatari, and I'm Professor of Sociology and Interim Dean of Interdisciplinary Programs at PLU. And Tamara and I are pleased to welcome you to COVID-19, a Global Crisis Examined, an interdisciplinary course hosted by PLU's Global Studies program and the Wong Center for Global Education.
In a year where both study away and issues-oriented public programming have been dramatically curtailed by a global pandemic, the Wong Center felt it was appropriate, perhaps even necessary, to create an opportunity for the extended PLU community to reflect on a global crisis that has upended almost every aspect of human life. Our health and well-being, our livelihoods, education, housing, global supply chains, our complacency around equity and access, to name only a few. To do this, we turn to our faculty colleagues and alumni, valued resources, who are not only living through the crisis themselves, but have also been engaged with it intellectually or professionally from a unique vantage point of their areas of expertise, and are able to provide a more nuanced perspective than what we receive every day in the media.
Over the summer, we invited PLU faculty to propose lectures that draw on their disciplinary expertise to understand the COVID-19 pandemic. Over the course of the term, we will learn from biologists, psychologists, political scientists, philosophers, anthropologists, scholars of religion, language, and theater, and from PLU alumni working on the front lines of medical care. By the end of the course, participants will understand different disciplinary approaches to the pandemic and be able to identify the social challenges related to the pandemic and various ways to address them.
We're genuinely thrilled to be offering this course to current PLU students, PLU alumni, community members, faculty, and staff. We're living in extraordinary times, and it is a privilege to be able to gather virtually to learn together. Thank you for being part of this. At any time, you're welcome to contact Teresa or myself to ask any questions.
And it's our great pleasure to begin the course with the inaugural lecture by two PLU biologists, Shannon Seidel and Eskew. Their lecture is entitled The Science of COVID-19, Part 1, The What, Where, and How of Coronaviruses. So I'll turn things over to Shannon and Evan to get us started.
Great. Thank you so much, Teresa and Tamara, for inviting us to be here today. It's an honor and a privilege, and I want to welcome everyone who's here. Alumni, students, staff, faculty, and community members, welcome. This is a really exciting course to be a part of. I am tasked with the job of giving you an introduction to the virus and the basic science of the SARS-CoV-2 virus. And I'm going to start to do that by giving you a little bit of comparison between the virus itself and us. So SARS-CoV-2, if you're not familiar, is the name of the virus that causes the disease COVID-19 that you are maybe more familiar with. And so if we take a look here, the genome of the virus actually only has about 29,000 base pairs. So when we think about that in comparison to our genome with over 3 billion base pairs, we can think how small this is, right? It's many more orders of magnitude smaller than our genome. It's that small. It only has about 10 genes as opposed to 20 to 30,000 genes in our genome. And yet it is wreaking havoc around the world and has disrupted every single one of our lives over the last 6 to 8 months. We consider humans alive. I think that's a pretty simple question, but it's pretty complicated to think about whether or not we define viruses as living. So I'm coming at this from a virology perspective as a biologist. And so when we ask the question of what is a living thing, we think about it often in terms of structure and function. This relationship between structure and function is actually a really important one in biology, and it's important for how we understand whether or not something is living. So to give you an example, a hammer is a particular structure. It's made out of a wood handle and a metal piece that is good at hitting nails into a wall. That's its function, right? A screwdriver has a very different function and therefore a different structure. So what I'd like you to do now in the chat window is to stop and think for a moment and maybe type into the chat window, what are some functions of living things that you can think of? So I'm hoping that you will help me out here, my community, and share your ideas in the chat window. And let's see what we come up with. What are some functions of living things? Okay, I'm seeing reproduce, procreate, and eat, respond to stimuli, respire, breathe, move, create oxygen. These are great answers, absolutely. Biodegrade, create and grow, digest, love. Beautiful, thank you. You can keep adding your ideas to this and I'll share some as well. So I appreciate the way in which you're thinking about life. And I'll show you some answers that a biologist might give. So a biologist might think of living things as things that can reproduce and develop, that can process energy, that can grow, and that can respond to stimuli. Many of the other things you listed in the chat window would also be functions of living things. When you think about functions of living things, though, you might think of exceptions to these rules, right? So there are certainly people we would consider living who maybe aren't able to reproduce either because they're early or later in their lifespan. So there are exceptions to these functions of living things that we would still consider living. So when we think about structures of living things, we primarily, since the invention of the microscope a couple hundred years ago, think about cells as the basic unit of living things. And in fact, there's a biological theory. The basis of cell theory is that organisms are made up of one or more cells, and that cells are the basic unit of life. So if a cell, a single cell, is the basic unit of life, are viruses alive according to this theory? You might put in the chat window what you think. Do you think they're alive or not? Are viruses made of cells? And I don't have time to check the window, but you can see what each other are predicting. In fact, viruses are not made of cells. So if you use a structural definition of life, you would not consider a virus a living thing. Now, as a virologist, I would consider them living on the functional definition, but we'll have to debate that later. So viruses are not made of cells. In fact, viruses are much simpler and often much smaller than cells. They contain a genome, like I told you before. They contain a protein coat, so some sort of structure that protects that genome. And some of them contain a lipid bilayer, some sort of membrane that surrounds the viral genome. And so this is the structure of a coronavirus, like the SARS-CoV-2 virus. And what you can see here is that there is a genome here in the middle. That genome in the case of SARS-CoV-2 is an RNA genome, and it's surrounded by some protein. So many of these colorful units around that are structured along the membrane here, like the spike protein, hemagglutinin, membrane, and envelope protein, are all structural proteins of this virus. I want to specifically point out the spike protein, which is this yellow one here on the outside, because it has a really important role in binding to the host cell and allowing the virus to enter a cell. So this structure, the spike protein, is really important for the function of this virus. And what I also wanted to point out is that despite how simple these viruses are, with only 10 proteins and one really small genome, these viruses are able to get into a complex host like ourselves and wreak havoc on our systems and our communities, right? So the spike protein, I want you to remember, because next week when you hear from Dr. Mary Ellard-Ivy, you are going to learn a lot more about how the vaccine development that is being done right now is targeting the spike protein. And you'll learn a lot more about how testing is often looking at the RNA, the viral RNA here. Okay, so those are going to be important to remember, so hold on to those. What I want to show you now is how the virus actually replicates within a host cell. So this is a basic function of the virus, and it depends again on its structure. So here you can see at the top of the screen the SARS-CoV-2 virus and those little tiny yellow spike proteins on the outside of the virus particle. And they bind to a receptor on host cells called ACE2. So the cellular ACE2 protein is here on the outside of the cell, and they help the virus get into the cell, deposit its genome, and inside of the cell, this virus replicates and makes many, many copies of itself that can exit the cell. Now, some of those copies might go on to cause disease within a single host, or some of those virus particles might either be coughed up or sneezed out or expelled via droplets or other particles as a person is talking, laughing, or singing. And that might be how this virus spreads from one host to another host. So what I really want you to know, you don't need to know all the molecular details of how this virus infects a cell, but what I want you to appreciate is this structure-function relationship and how viruses, despite being so simple, can go in and hijack and use many important components of the cell's environment, making many copies and leaving the cell. Another thing, because I do want you to leave today with an appreciation of viruses and virology, despite all of the problems they're causing in your life right now, is that living things, all living things, from bacteria to humans to trees to mushrooms, all living things that are made of cells have one type of genome. It's double-stranded DNA, right? So you've probably seen that double helix. That is the genome type of all cell-based life forms. But viruses, on the other hand, have so many different kinds of genomes. And this virus is one example. So you can see viruses can have a single-stranded DNA genome or a double-stranded DNA genome. It can be plus or minus. It can be RNA. It can be double-stranded RNA. So there are just many, many different types of genomes, and viruses can infect every single living thing. So the diversity of viruses is far more vast than the diversity of every life form on Earth. It's really amazing and quite powerful to understand viruses and use them as tools. So viruses, despite this one being quite harmful, can do a lot of good in the world as well. And finally, before passing things on to my colleague, I wanted to show you how much powerful information we're learning about this SARS-CoV-2 genome. So I told you that the RNA, it makes up the genome, and it's about 29,000 base pairs. What you can see along the bottom of this Explorer from nextstrain.org is that we have sequenced the whole genome of the SARS-CoV-2 virus many, many times over. In fact, right now, this is showing, as of today, about 4,400 copies of sequences of this virus from different people all around the world who've been infected. And what we can learn from this is about how the virus is changing and mutating over time. So we're finding specific areas where there are high mutation rates in the virus, like here, and other areas where the virus is really consistent. This can have implications for treatments, for therapeutics, for vaccine strategies, and much more. So we're learning a lot about the virus every day, and much more information has been shared about this than any virus we've studied ever before in such a short time. So with that, I'm going to pass things off to my colleague, and I'm going to come back at the end to talk about the disease triangle. So stay tuned for Dr. Eskew.
All right, everyone's seeing my screen now. We're good to go. Can you confirm, Shannon?
Yes, I'm seeing your screen.
Awesome. Hi, everybody. I'm Evan Eskew. I'm an assistant professor of biology here at PLU. And so Shannon introduced you to the virus, SARS coronavirus 2. She talked about its structure and how its structure is intimately related to its function. In particular, it being able to invade and replicate within host cells, within the cells of a human host in particular. What I'm going to talk about is to step back a little bit, to kind of broaden our lens a little bit, and to think about how SARS coronavirus 2 would be introduced into the human body to begin with. And this slide I have here as my background, hopefully is giving you some hints and is leading the way to the rest of this mini lecture. So we believe as the scientific community that SARS coronavirus 2 is what's known as a zoonotic pathogen. So a pathogen is a organism that, or a thing that can cause disease. And in particular, SARS coronavirus 2 would be a zoonotic virus, right? That makes COVID-19, the disease caused by SARS coronavirus 2, a zoonotic disease, also known as a zoonosis. So using a lot of these big words that begin with zoo, what is a zoonotic disease? That's just our term for a disease that has jumped or can be transmitted from a non-human animal, an animal in common parlance, to a human host. So zoonoses come to us from animals. And that process of transmission across host species from one biological species to another is called spillover. So we would say that the pathogen, in this case the virus, spills over from some animal host or a community of animal hosts into people. And so to get everyone involved, we've had great participation already. I would like us all to brainstorm a little bit. And again, hopefully this slide gives you some hints. So take a look before I go to the next one. I would like you all to brainstorm some ways in which you think or you suspect or you know that a virus could spill over from animals to people. So another way to put that same question is, how would a virus get from animals to people? Okay, we've got a lot of ideas. We've got a bite, animals biting, food, if they eat the animal, ingestion, poop. I'm gonna try to condense these some. So yeah, so we have a number of these that are related to eating something, or perhaps being contaminated by another, by some part, I should say bodily fluid of the animal. So we have guano, we have feces, so coming in contact with the pathogen in that way. We have water contamination, mosquito bites, and that was further up as well. So we had a number of animal bites slash mosquito bites, inhalation, ingestion. Thank you all. I'm scrolling through because they came in fast. All right. Any other takers out there? Do you think you have the pathway that someone else has not said yet? Please feel free to weigh in. Contact with self and self-inoculation. Yeah, so we've got a lot of great ideas, and you guys you're doing my work for me because you've covered most of the things that I was going to talk about. Anyone else out there, if you've got your fingers on the keyboard, go ahead and type it. Petting an animal, kissing, yeah, being in close contact with perhaps a pet in that way. All right, awesome. You guys can keep adding your ideas as I talk, but let me continue on. So zoonotic diseases, as you guys have shown already, can be transmitted in a number of different ways. So this infographic puts these different types of contact, these different transmission pathways, we might call them, into some broad categories, many of which you mentioned already. Air and waterborne transmission from a vector, so like a mosquito, proximity and direct contact with animals, and food, which also many of you mentioned. So I'm going to kind of speed through some of the next slides that I had, simply because you've already hit on these, and you know about some of these things. It's apparent from your answers. So first, yeah, one of the more obvious ways that zoonotic transmission can happen is through direct contact with an animal. So for example, a raccoon or other mammal that's infected with rabies, also a virus, can bite a human and therefore spread rabies in that manner. And I would say that as ecologists and disease ecologists often think about vector-borne transmission as a different category. So in that case, a mosquito or perhaps a tick is basically vectoring, they're bringing the pathogen from one animal host to humans, but it's kind of a very similar mechanism, right? A bite, a direct bite is what leads to infection. There's also the potential for disease transmission because of food and food-related activities. So here we have an image of a wildlife market. I believe it's Indonesia. You see a number of different wildlife species for sale in this market. We have some lizards, some sort of small mammal, a rodent of some kind, and then further to the left there, perhaps another larger mammal. So in the process of hunting animals, butchering them, bringing them to a market, and consuming them, all of those different activities, there's the potential for disease transmission. And sorry, I think I skipped over that a couple of slides ago. I would just say that the easiest way to think about it is almost any human activity that brings humans into close contact with animals has some potential to cause, or carries some risk, I should say, of zoonotic disease transmission, obviously depending on the activities and the animals involved. But so, as I said, there are a number of these different food-related processes that can be involved, and they're not restricted, I would make note of, to a market like this that might be selling wild animals. Industrial scale farming in places like the US also carries that risk, right? So if you have livestock or poultry in extremely high densities, those are animals, and those are animals that can carry zoonotic pathogens, and they can transmit them to humans who work around them or consume them. So those are kind of two, the direct contact and food related, I think, are two that a lot of people have heard about, have some degree of knowledge about. I wanted to point out a couple of examples that I thought were interesting and maybe you have not heard of. So there are more indirect ways that people might come in contact with animals, or in particular their bodily fluids. So what we have depicted here is a date palm tree. So date palms are widely distributed globally now. They are the tree that we get the date fruit from, but they also have a sap that people like to harvest. So a good analog is like a maple tree. So maple trees are tapped. You create a hole in the tree to extract the syrup, and people do a very similar thing with date palm trees. So the sap, as you can see, is collected into these buckets through a little funnel, and this sap is consumed directly as a food item. It's also fermented, I believe, to make alcoholic beverages in some regions. People want this sap, and they collect it in this manner. Unfortunately, humans are not the only animals that want this sap. So here's a nighttime image of a bat that's coming along to this date palm tap and licking along the very place in the tree that humans are trying to collect sap from. And what can happen, this particular activity, date palm sap collection, has been linked to Nipah virus transmission in Southeast Asia. So Nipah virus is an extremely deadly virus. It's in the Hendra virus family, I believe. And bats essentially come, they can either contaminate the date palm containers, the sap containers, with their urine or their saliva. And when people consume that contaminated food item, they can become infected themselves. Another method of indirect contact, which actually people did hit on, I was a little bit surprised that people were familiar with this one, is activities like bat guano harvesting. So you might have never thought about the fact that people around the world in certain regions harvest bat guano for use as a fertilizer. So when you don't have access to kind of industrial, fertilizers, you can collect guano from animals. Collecting guano involves going into habitats that those animals occupy. So in particular, a bat cave or some sort of similar habitat. So obviously in the process of collecting these animal products, you also carry some risk of zoonotic disease transmission as well. So thank you everyone for those ideas, for contributing to the discussion. So we now have some sense of how a pathogen, a virus, specifically might move from animals into people. But why do we really care that much about zoonotic diseases in general? Well, here's one reason. So here's the kind of the main figure, the main takeaway from a paper that came out in 2008. What these authors did is they actually tried to identify every emerging infectious disease event since 1940. So you could think of these as disease outbreak events. So they identified these events from the literature and collected data on what pathogen, what sorts of activities were involved. So characteristics of the disease outbreaks. And you can see their data here. So first of all, we see that they identified increasing numbers of infectious disease events over time since 1940. And in addition, the colors of these bars are really important. So any color that is not black indicates a zoonotic disease. That EID event was a zoonotic event. So you see that about 60% of these spillover events, or sorry, these disease events were zoonotic. And of those zoonotic events, about 70% of them come from wildlife. So it's not just zoonotic diseases that appear to be pretty prevalent and important, but in particular those that come to us from wildlife. And zoonotic diseases are really many of the ones that you've probably heard of most commonly. So HIV, originally a zoonotic disease, the precursor virus to HIV, we think spilled over from primates into humans a number of decades ago. Ebola, bubonic plague, some forms of flu, rabies has already been mentioned, Lyme disease, hantavirus, Nipah virus, Hendra virus, Zika virus, West Nile virus, toxoplasmosis. Not all of those were viruses, but they all are zoonotic diseases. So some of those caused by bacterium. All of those have wildlife or some sort of animal host that we often refer to as a reservoir. So the reservoir is where the pathogen is hanging out in a kind of a natural setting before it might spill over into humans. So why do we think that SARS coronavirus 2 in particular is a zoonotic virus? Well, many of you may be kind of familiar with the general outline of this story, but very early on in the COVID-19 outbreak globally, there was a lot of attention around the Huanan seafood market. So this particular location in China where, as you see Bloomberg News here on January 20th, there was the suspicion that the virus had spilled over into people at this food market. So a number of different wildlife items were for sale in this market, and some of the initial cases of COVID-19, people appear to either work or have gone to this market. So obviously that sort of sets the expectation or the idea that perhaps this pathogen is a zoonotic pathogen. I want to specifically take the time now to kind of reverse that narrative. So everything's happened so fast with COVID and we get bombarded with the news all the time. A lot of things get lost, and some stories are particularly sticky in people's minds and persist when perhaps they shouldn't have that staying power. So this article is from January 20th. January 26th, 6 days later, in Science Magazine, a popular summary published in Science Magazine is this article by John Cohen. He's actually talking about original research published in the medical journal The Lancet, which was describing a set of initial clinical cases of COVID-19 in China. So a total of 41 cases. They have information on the patients, what they were doing, their age, all these sorts of things. Really interestingly, the earliest case in this cohort, the earliest person to become ill, had no known contact with the seafood market at all. In addition, 13 of those 41 cases had no link with this seafood market. So from these data, it seems very plausible that rather than the spillover event happening at the market, humans were already infected and they happened to come at the market, or there was human-to-human transmission perhaps in the market setting, but not necessarily spillover originally happening in the market setting. So that being the case, why would we still think SARS coronavirus 2 is a zoonotic pathogen? If it didn't obviously come from the market, why would we think that? Well, really it's because we have precedent with other very closely related coronaviruses. So first of all, we have the case of the original SARS coronavirus. So SARS stands for severe acute respiratory syndrome. That's the disease. And the virus that causes this disease, we just call SARS coronavirus. So that's why SARS coronavirus 2 has to be called SARS coronavirus 2. It's the second one. The initial spillover of SARS happened in late 2002 in South China, and it resulted in international spread of this disease. You can see here a depiction of the case counts in different parts of the world. It resulted in about 8,000 cases, 800 deaths, but there have been none, as far as I know, since 2004. Now, really interestingly, we think, tracked down the natural reservoir, the animal host of SARS. So SARS is also, we now know, to be a zoonotic virus. And the natural reservoir of SARS appears to be horseshoe bats. So horseshoe bats are a group of bats that are widely distributed throughout what ecologists would call the old world, so Africa, Europe, Asia. There's a species of horseshoe bat that ranges from Spain to Japan. So they're extremely broadly distributed throughout that region. But it took us a really long time, I say we, as the royal we, the scientific community, to actually understand what was going on with SARS spillover. So I wanted to particularly highlight this paper from 2017 from a group of Chinese scientists. What they essentially do is they went into bat caves, they sampled coronaviruses from bats, and what they found is, as you can see underlined, essentially all of the building blocks of the original SARS. So they didn't find SARS itself, but they found all the pieces, all the genetic pieces needed to make SARS. So this suggests that bats are the natural reservoir. I also wanted to highlight the timing of this. SARS outbreak happens in 2002. It's 15 years, almost to the month, 15 years to the month, that we have a pretty good idea of where we think that this virus came from. It takes a really long time. I also wanted to point out that all of the research that happens around coronaviruses since SARS basically, over the last decade plus, have kind of been warning signs about SARS coronavirus 2. Here's an article from The Guardian published in 2017 about this paper. And they say that the Chinese team warned that another deadly outbreak of SARS could emerge at any time. As they point out, the cave where they discovered their strain is only 1 kilometer from the nearest village. So obviously there's a number of pathogens out there that can do us harm, but coronaviruses in particular have been high on people's watch list in the scientific or the disease community at least. In addition, we have another kind of example in MERS. So MERS, you may have heard less about, Middle East Respiratory Syndrome, a respiratory disease that was first identified in 2012, also caused by a coronavirus, also appears to be a zoonotic disease. It has caused fewer cases than SARS, only about 2,500 cases, but resulted in the same number of deaths, about 800. So it actually has a higher case fatality rate than SARS. It's more deadly given infection. And there have been infections in 27 countries, pretty major outbreaks in Saudi Arabia and South Korea. Interestingly, as this little infographic on the left-hand side of the figure suggests, a lot of transmission appears to happen through camels. So in particular in the Middle East, where camels are used as work animals, camels appear to transmit MERS to people, and then people can transmit it amongst themselves. But analysis of the coronavirus genomes, of the MERS genomes, so looking at the physical structure of those viruses, suggests that the evolutionary source of MERS is actually also in bats rather than camels. So there's some sort of history of events of bats giving the pathogen to camels, which then spread it to people, it seems most directly now. So we have these two precedents for why SARS coronavirus 2 might be zoonotic, and we also have direct evidence from the structure of SARS coronavirus 2 genome itself. So here's a paper that came out in Nature in February, and the kind of primary results of this paper. So what you see in the figure along the x-axis, along the bottom, that's essentially looking at the length of a coronavirus genome. So if you were to take the genetic material and stretch it out, it's like you're looking at those base pairs throughout the entire genome. And what the y-axis is showing is how similar a given virus is to SARS coronavirus 2, so collected from humans. What you see here, these 5 different colored lines represent different coronaviruses and how similar they are to SARS coronavirus 2. So you have the original SARS in the red line. And if you trace that red line, you see at some parts of the genome, it's about 80% similar to SARS coronavirus 2. In other parts, it's much lower than that, around 50% similar. But the big takeaway from this figure is that blue line. That blue line is a coronavirus called RATG13, not a particularly helpful name or a memorable name, but it is a coronavirus collected from, guess what, a horseshoe bat that currently has the highest known similarity to SARS coronavirus 2. So it's the closest relative, again, that we currently know of to SARS coronavirus 2. It's not SARS coronavirus 2, and it's actually not extremely close in terms of ancestry. I read some estimates that were about 40 years. So this closest known virus probably diverged from SARS coronavirus 2, the one causing COVID-19, about 40 years ago. But again, the kind of collection of evidence suggests that some sort of wildlife is probably implicated. Bats make a lot of sense based on what we know about SARS, and we have this evidence of very closely related bat coronaviruses circulating in the wild. So where does this kind of leave us with understanding where SARS coronavirus 2 came from? Well, to be honest, I want to say that we might never be able to know precisely, and people kind of resist that. People obviously have an interest in knowing one clear-cut answer, but that event is already months, potentially more in the past from this current moment. It's going to be very hard to track down the initial case, the initial person, the initial animal or set of animals and region where this spillover event may have occurred. But doing viral surveillance in wildlife can get us closer to understanding some of these issues. So, right, in that previous slide, if we had never sampled for coronaviruses in bats, we would never know that the most closely related virus to SARS coronavirus 2 came from a bat. So doing general viral surveillance in wildlife helps us understand the diversity of those pathogens in wildlife right now. And then in addition, doing surveillance in humans, and I would especially say serosurveys. So surveying for antibodies, which are actually a signal of prior infection, could be really important. Because there could be regions, communities, people in certain industries that are actually at high risk of spillover, and they don't perhaps get symptoms from the pathogens they get. Maybe they misattribute those symptoms. You get sick from something, you thought it was a cold or you thought it was flu, and it's actually potentially a novel pathogen. So doing that sort of basic scientific groundwork could show us where and why are people potentially being exposed to viruses that could be a real problem for us. And with that, I'm going to turn it back over to Shannon to finish up our lecture.
Okay, thanks for sharing all of that. So we have an activity to have you do to kind of bring together these biological perspectives of virology, as well as ecology and understanding community transmission. So if you could go to the next slide, Evan, we're gonna set up that activity. So get ready to do a little bit of work. And this is all based on an epidemiological concept called the disease triangle. So any given disease that you put in the middle is impacted by the pathogen, specifically in this case, the virus, the host, so in this case, individual humans, and the environment. And the environment can be everything from how humans interact with one another, cultural practices, the climate and weather, many other things. So what we're gonna have you do now is think about the disease COVID-19, and if you can just tap that one more time, Evan, we're going to have you use the Google Slides link that I am now putting into the window. And if you click on that link from the chat, you should be brought to a Google Slides document that has many copies of a slide that looks just like this disease triangle. And I want you to go to a slide that only has maybe 2 to 4 people on it. So if you go to that slide, as you can see Evan doing now, you can find a slide with just a couple people and add your ideas. What are some pathogen-specific factors? What are some host-specific factors? And what are some environment-specific factors? What we think this is going to allow you to do is to think about this biological discipline and also a lot of the other disciplines you're going to think about over the rest of this course. Because especially host-specific and environmental factors are going to be impacted by psychology and by political science and by many other aspects, right? Ethics and government decision-making and many other things. So let's go see what kind of things you're adding to that document. There's many copies, so feel free to spread out and find a slide to work on. We'll give you about 3 minutes to do that, so just think a bit. And so once you're there, feel free to just tap on the slide and type in your pathogen-specific factors, your environmental factors, or your host-specific factors. Feel free to add your ideas there. This is great. Y'all are doing a great job.
So Shannon, someone in the chat just asked if you could define pathogen in a different way.
Sure. Oh, thank you. So when we're talking about the pathogen, in this case, we're talking about the virus SARS-CoV-2. So the virus that I showed you earlier that had an RNA genome, a bunch of proteins, and a lipid bilayer. So things that are specific to that little structural unit of SARS-CoV-2. Thanks for that question. So I'll give you about a minute to come up with a few more ideas in this brainstorm. Y'all are doing a wonderful job. Like Evan said, you're doing the work for us. And now just take a look at some of the other slides. So as you are finishing up your ideas, you might look at a couple of other slides and see what other people came up with. And you can see there are so many great ideas. And so if we circle back, I think we could probably spend more time if we had it, but we want to make sure we save time for some Q&A at the end. So if we circle back to our Zoom screen share, and if Evan clicks to the next slide for me, thank you. Here's some ideas that we came up with to share with you. And many of the things that you put in the slide document there were there as well. So in terms of the pathogen, so there are factors of the pathogen that specifically impact this disease, right? If the mutation rate of the virus, the route of entry of the virus, how it gets into a human host and what cells it infects initially matters, the structure of the pathogen, all of those things. But virology alone and understanding the pathogen can't help us really fully understand COVID-19, the disease, because host and environmental factors impact that as well. So as you're starting to think about the course over the next 13 or 14 weeks, you can think about how these factors fit into other disciplines as well. So mask compliance, physical distancing, some of these have to do with the ethics of what is being regulated and what is not. So natural disasters that are happening around the world are impacting how close people are to one another as they're being evacuated from homes, even in our own communities right now. Testing availability and healthcare availability are important environmental factors. And then of course, there's many host-specific factors that include genetics and age. Race and ethnicity has been shown to be an important factor in this disease. Immune response, compliance with hand washing. There are so many factors that are involved that involve human behavior, community interactions. And so this disease triangle, which is a concept from epidemiology, really shows how COVID-19 is an interdisciplinary problem and is going to require an interdisciplinary solution. So I really look forward to taking the rest of the class along with you all and hearing from my colleagues from a lot of other disciplinary perspectives. So with that, we will say thank you so much for your time and for your participation and your energy today. And if you would like to ask some questions, you can start putting them in the Q&A. So we'd prefer that the questions here now come from the Q&A rather than the chat window. So there's a Q&A button there at the bottom of your screen. And we'll give you a moment to think of questions you might have so we can dig a little deeper. And maybe Evan, you want to join me back on screen here?
Yes.
And we'll see what questions come up from all of you. Or maybe you're still doing the disease triangle activity, typing in the document. So if anybody has questions, we are happy to answer.
Can you see Shannon, or would you like me to read?
Oh, I see one here. Okay. So the question is, Dr. Seidel, you mentioned that viruses are not only harmful. What are some examples of positive functions of virus? Thank you for the question. I love to talk about benefits of viruses. So a couple that I'll mention are gene therapy. So a lot of gene therapy options use viruses to deliver genes to cells, because viruses are really good at delivering genes to cells, right? So we actually can co-opt and use that for a beneficial purpose. You'll hear next week about from Dr. Mary Ellard-Ivy about how viruses are used to vaccinate people. So that's a benefit. And then another one is just for understanding cell biology. A lot of what we know about how cells function is from viruses hijacking them, because viruses tend to go in and hijack the most important parts of the cell. So a lot of what we understand about how cells replicate, how cancer is caused, things like that are from learning about viruses. Those are some benefits. Wow, lots of questions coming in. Thank you. Evan, is there another one that you'd want to answer?
I can take one.
Great, go for it.
That is more my speed. There's a lot of questions about transmission that I'm not sure I'm qualified to answer. But one that I can is from Aaron McKenna. How do we stop people from seeing bats as bad? That's a significant debate within the conservation community and folks who do bat research in general. So bats themselves are right now impacted by a significant infectious disease that's caused by a fungus called white nose syndrome. So bats are impacted by disease, and they also play this significant role in disease transmission to people. One thing I would say is that, first of all, bats have a lot of positive functions. So as Shannon was just talking about positive functions of viruses, bats give us significant ecosystem services in terms of pest control, for instance. They are free pest control that is, it's worth a lot of money. So there have been studies who did this. They try to add up how many insects do bats potentially kill, you know, in the Midwest on corn farms and that sort of thing. How much is it worth? It's many millions of dollars, if not more, worth of pest control we get from get for free, basically. So that's one answer, is that to emphasize their positive attributes. And the other is that viewing bats as bad or trying to eradicate bats is often a very detrimental strategy that works against disease prevention. So in cases where people try to go out and cull, they persecute bats to try to kill them, that can actually increase stress, increase movement of natural populations, and increase the risk of disease transmission to people. So really at its core, it's about learning to coexist with bats, and many of the activities that result in disease transmission from bats are things where basically we're invading their space. So I think that those kind of sets of answers get us more progress than just seeing bats as bad, quote unquote.
Great. I can take a couple of the transmission questions that are somewhat related. So one of the questions is about how we can explain asymptomatic individuals. And the disease triangle is a really great way to think about that, particularly factors that are both host and environmental specific, although some pathogen specific ones as well. So what we know about SARS-CoV-2 is that because we humans get infected by other coronaviruses, there are a couple coronaviruses that cause the common cold that have enough similarity in sequence that there is some immune response in some individuals that is at least maybe partially protective for SARS-CoV-2. So this virus is novel, but we are starting to find out a little bit more about the immune response, and there is some protection in some cases from individuals. So those people, that would be my hypothesis. I don't think we have evidence on this yet, but that asymptomatic people have some partial immunity from other coronaviruses. That would be one explanation. Or that there are other immune specific response because of genetic differences between humans that allow people to fight off the virus more efficiently, or because of the route of transmission, or because of the amount of virus that a person is infected with. Those all might be possible explanations for symptomatic, asymptomatic individuals. And then there's a related question about reinfection, right? So after someone has been infected, they typically develop an immune response, and that immune response, we hope, becomes protective. We're starting to find cases of reinfection. There are a few that have been documented and shared in the literature. But for the most part, those have resulted in lesser symptoms the second time around. So there have been unique viruses sequenced from an individual over time, but the person was more sick the first time around and less sick the second time around. So that doesn't mean that will always be the case, and that will be the case in every situation, but that is at least a hopeful sign. So let's see.
Evan, I think you're muted.
Oh.
It had to happen to somebody, didn't it? I was going to say that there's a lot of questions coming in, so if we don't get to everyone, don't feel bad. There's a lot of stuff in here, some of which I'm not sure myself or Shannon would be qualified to answer, or at least speaking for myself. But I have one here that is interesting. Kenny asked, why has SARS coronavirus 2 been so much more devastating than SARS coronavirus? I think that's really interesting because I read some things recently that kind of suggest that we may have gotten lucky with control of SARS coronavirus. So it did spread internationally, but in places like Canada, where it spread to, other parts of Southeast Asia outside of China, it actually got controlled relatively quickly, and not necessarily, the things I've read suggest there might have been a little bit of luck there. I think the other really big part of SARS coronavirus 2 is this asymptomatic transmission, right? So when you have a disease, let's say like an Ebola or a Nipah virus that has a lot higher case fatality rate, in a way, controlling spread is a little bit easier, because if you can identify people who are infected, then it's easier to do control, right? So with SARS coronavirus 2, a significant proportion of people aren't showing symptoms, it's very difficult to find out that they're infected and to prevent further spread for that way. So that might be a partial answer, and maybe Shannon has others.
No, I think that's great. There's so many questions that I hope we can get to, but so I'm just going to start with, are any of the current vaccine treatments looking to leverage CRISPR in innovative ways that are different from traditional vaccine development process? I want to just plug Dr. Ellard-Ivy's talk next week, because she has expertise in CRISPR and we'll be talking about vaccines and treatments. So stay tuned for that one. And then there's a question here about the amount of it, what we mean by how much virus gets into a person and how that is actually measured. That's a pretty easy one to answer, is that we're talking about how many infectious virions or individual virus particles in that case. And so one of the fundamental virology questions that we don't have a great answer to for this or for other viruses in many cases is how many virus particles have to enter a particular host in order for an infection to occur. It's not one. It's not one virus gets into my body and I become infected with SARS or with COVID-19. It may be in, people have talked about 100 or 10,000 virus particles, right? So there's a huge range and we don't actually know the answer. So that's a really interesting fundamental question, but we would measure it by the number of infectious particles.
And that that question is really interesting. It's essentially the same process. So PCR is the technique that's used to do testing, which you'll probably hear more about next week. And we use that technique widely in biology. I use that technique to sample fungal pathogen from amphibians. So swab their skin, run a PCR to detect the DNA of the pathogen, and also to quantify it. So there's also some really interesting things I've read recently where, for the most part in COVID testing, if it's done by PCR, we're just reporting, were you infected or not? So we take a continuous measure. There is a measure of how much virus roughly there was, and just create a cutoff where if it was over this threshold, we say the person was positive, and if not, they were negative. But there are people advocating, why are we throwing this information away? Because it could be really important to know how much virus they had at the time they were tested. And indeed, it's important for almost any disease we're studying. I care, is the frog really infected with fungus or barely had any? And similarly, you would think we would want that information for COVID patients as well.
Yeah. Yeah, and that gets at some of the, there's another question here about the evidence that there's transmission from students that are younger than 14 to adults like teachers, for example. So should schools be open, right? And the answer is that we don't know super well yet. We have different examples and case studies from around the country and around the world, but it has a lot to do with the viral load. So how much virus is being produced in that younger than 14-year-old student, whether they're symptomatic or asymptomatic, that they can pass to another person. And so it would depend on many of the host-specific factors that we mentioned in the disease triangle, right? So it is really complicated, and we're still learning a lot.
Yeah, I can do another one, interesting one. So why is the mutation rate higher in some areas as compared to others? So I think that's probably based on that plot with the different coronaviruses and differences in similarity across the genome. Well, as a basic fact, there are parts of the genome for humans and for many other organisms that are more important than others. There are certain genes that are absolutely critical to your development as a human being. And if you have mutations in those genes, you don't have a viable embryo, for instance. So you can't have mutations in certain areas that are critical in that way. But there's large swaths of the genome that we aren't necessarily sure the function, or appears to be a lot more flexibility in terms of the fitness impacts of mutations in those regions. So I think that would essentially be the answer for viruses as well. There's stretches of the genome that code for proteins or not, that simply are viruses can survive and they have some success with mutations in those regions, whereas other regions are highly canalized. They have to be, very well conserved over evolutionary time.
Right. And those are usually important for the structure, for example, of a particular protein. Yeah. Yeah. So,
So excuse me, Shannon. I think we have time for like one more question.
Oh, there's so many. Somebody's asking about the specific factors of the pathogen that might be the most important for the potency of the virus. And it really does depend, right? It depends on a lot of factors, and it's something that we will learn over time. So that nextstrain.org that I showed you, that's what a lot of people are looking at right now, is to try and figure out which mutations stick and persist in the population, because they probably have some advantage over evolutionarily. Great questions. And so many more that we're not getting to. Somebody asked if we'd share the slide deck, and I'm happy to do that. And I think that we can make sure that gets sent out with the video and shared on the Sakai site for students. Should we call it there?
Yes, I think so. It's been so wonderful. Evan and Shannon, thank you very much for just setting us on the right track. It's been wonderful. I've gotten several messages.
Thank you so much for the opportunity. It's been great to be here with you all.
Great. Thank you so much. And we are at time. So we really, really appreciate how you've sort of set us on the right track. And we look forward to next week when Mary Ellard-Ivy will continue on the biology track with her talk, The Science of COVID-19, Part 2, Testing and Vaccine Development. If you want to share the slide deck with me, we'll make sure, Teresa and I'll make sure that it gets sent out with the recording. But for now, thanks to everybody that joined us. It's been a delightful evening. And unfortunately, we have that really sudden, very awful cutoff that we have to do with Zoom. There's no like hellos and how are you and see you next week. So we're going to have to sign off soon. But thank you so much, Shannon and Evan. And Teresa, we'll see you next week.
All right.
Take care, everybody.
Bye-bye.